so we've spent a lot of time up to this point talking about the plasma membrane, its composition, that it's made up of phospholipids and proteins. We've talked about the different roles of proteins, and we've talked about kind of how we get things across that particular kind of membrane. We need to move on and talk a little bit more about the other stuff found within the cell, including the cytoplasm and stuff that's found along with the cytoplasm. So let's kind of move in that direction next. Recall that the cytoplasm refers to basically all the stuff inside of the interior of the cell, but not including the nucleus. So anything that's outside the nucleus but inside the plasma membrane is technically cytoplasm. The cytosol, we said, was the fluid component of this cytoplasm. So in other words, it's sort of the water part of it. And then we have cytoskeleton, which is a series of protein structures that are going to produce a scaffolding inside of the cell membrane that supports the cell membrane and gives the cell a certain shape. This cytoskeleton is going to be made up of a variety of different types of protein. We have these large proteins known as microtubules. In the image on the left, you see a short example of a microtubule. It's formed like a string of beads that is wrapped around in a coil. That's a microtubule. Then below that you'll see these microfilaments, also known as intermediate filaments. These are sort of, they look like those ropes of licorice that kind of are bundled together, and you can kind of peel off one little strand at a time. And then you have, or I'm sorry, those were, that's the intermediate filaments. They're that way. The microfilaments are the small ones, also known as actin filaments. So you kind of, going from largest to smallest, it goes microtubules, intermediate filaments, and then microfilaments or actin filaments. Okay, and remember again that the cytoskeleton is kind of a structural component that gives some shape to the cell. Next, let's take a look at the organelles, and we're going to move through these relatively quickly. These are small structures within the cell that have a very specialized function. They do some specific function for the cell. And the main thing that you need to understand from this is just recognition of the names. You certainly aren't going to be expected to know absolutely everything that each centriole does. So basically I want you to recognize the name of the organelle and then just a very, very fundamental role that organelle plays within the cell. So the first one is something called a centriole. Here's an example of a picture of what a centriole kind of looks like. These things are anchor proteins within the cell that are probably play a role during cell division. They become an anchor point for other structures during cell division. So if you can just remember that centrioles have this kind of an appearance and that they have a role in cell division, that's probably enough to know for a centriole. Cilia, these are technically, they can be considered organelles, but they're also specialized extensions of the plasma membrane. So you guys saw examples of cilia in pseudostratified columnar epithelium, where we saw these very long finger-like protrusions of the membrane. Well, this is what they are. They're these long whip-like structures that are covered by plasma membrane, but on the interior they're supported by these long microtubule arrangements. And because of the way these things are set up with microtubules, ATP acts on the microtubules, causing them to shift relative to one another, and that shifting of the microtubules causes the cilia to whip back and forth, and this produces a kind of current in the extracellular fluid surrounding the cell. You can see an example of this idea of the whipping in this lower image on the right. An example of a very specialized type of cilia, what might be like the flagella component on the end of a spermatozoa cell. So the flagella, the long tail-like component of a sperm cell, would be an example of a single cilia. Structurally, they're the same, and they can kind of whip back and forth and propel the cell forward. The next structure is a microvilli. Here you saw again the microvilli, also known as the brush border. Those were the tops of the simple columnar epithelial cells you saw in lab. These are very short folds in the membrane, and the cytoskeletal structure inside that supports them is these small microfilaments or actin filaments. And here you can see those small filaments forming a kind of gridwork or scaffolding inside the microvillus to support that particular fold. Unlike the microtubules that form the structure of a cilia, the microvilli and the actin filaments that support them, those do not move. Those do not change, and they don't cause the microvillus to stroke back and forth. They're just a stationary kind of structure. So they're kind of an extension of that plasma membrane. I think I might have mentioned this before, but the role of the microvilli is to increase the surface area of the cell. And particularly in the simple columnar epithelium that you guys saw in lab, that particular kind of epithelium lines the digestive system. And the microvilli is heavily populated with digestive enzymes that are bound in the cell membrane. And so by folding up the membrane like this, it increases the amount of cell surface that you have, and therefore increases the amount of digestive enzymes that you can put within a specific region. I've already mentioned that they're smaller than cilia and they do not move. Okay, next organelle is a ribosome. We'll learn more about ribosomes a little bit later on, but on this image here, the ribosomes represent the small blue dots. Ribosomes are an important organelle for the production of proteins. We'll talk about that process, but for right now, just know that ribosomes play a role in protein synthesis. Endoplasmic reticulum, in the cartoon image, you'll see a gray representation of a cell, and notice that the little round blob in the center, that represents the nucleus, but notice that it's surrounded by this colored region that's kind of blue and pink. That structure surrounding the nucleus is known as the endoplasmic reticulum, and it's a very elaborate membrane structure that plays two important roles for the cell. Parts of this membrane are going to have a large number of ribosomes attached to the membrane, and because of the attachment of these small little ribosome proteins, when you look at the endoplasmic reticulum through a microscope, it makes the endoplasmic reticulum look very rough, like it has rivets or studs attached to it. And so rough endoplasmic reticulum is the type of reticulum that has a lot of ribosomes attached. And since you already know that ribosomes are involved in protein synthesis, you can assume that the rough endoplasmic reticulum is a region where a lot of protein synthesis is occurring. There are portions of the endoplasmic reticulum that do not have ribosomes attached, and because of that, it doesn't have that studded or riveted appearance and is consequently referred to as smooth endoplasmic reticulum. And this is a place where the cell is generating lipids, particularly phospholipid membranes. It's a kind of phospholipid membrane factory. Okay, the next organelle is the Golgi apparatus. This is a center of the cell that is involved in packaging and processing proteins that have been made. So if you think of the interior of the cell as being like a little city or a little factory, proteins are going to be made in the endoplasmic reticulum, and many of those proteins, once they've been produced, get moved from the endoplasmic reticulum via vesicles to the Golgi apparatus. And within the Golgi apparatus, they are folded up into their final form and shape, packaged into small vesicles, and then those vesicles are moved toward the plasma membrane and released outside of the cell through exocytosis. So the role of the Golgi apparatus, again, is kind of processing of proteins and packaging those to be shipped out of the cell later on. Here's kind of an example of that, a summary showing how that would work. In the upper part of the image, what you're looking at is rough endoplasmic reticulum. So it's a membrane structure with ribosomes attached to it. The ribosome is involved in producing this small protein, which is this little blue bead-looking structure that's forming in the interior of the endoplasmic reticulum. Once the protein has been formed, the endoplasmic reticulum pinches off and forms a vesicle. Then if you look down into the lower part of the image, you'll see these vesicles being moved toward the green structure, which represents the Golgi apparatus. The vesicle becomes part of the Golgi. The protein is released into the interior of the Golgi apparatus, and as it moves through the Golgi folds it up into its secondary and then tertiary and maybe even quaternary form of the protein, and then once it's completely folded it correctly, it forms it into another vesicle, shown here by number 5, and that vesicle is then moved to the toward the surface of the cell. As we see in number 7, and through exocytosis, that protein is now released outside of the cell. One of the things that the Golgi may make are structures called lysosomes, and these are actually another type of organelle of sorts. And these lysosomes, their role is to actually digest structures within the cell. So here you see a lysosome. It's basically a vesicle that contains digestive enzymes. And if the cell takes in some kind of vesicle containing a substance from outside the cell, this lysosome will be attracted to the vesicle containing the new substance. The two vesicles will fuse together, and the lysosome's enzymes will digest or break down the components inside of the vesicle that was brought in through endocytosis. Finally, we have peroxisomes and proteasomes. These are smaller versions of lysosomes that do specific kind of actions like break down fatty and amino acids. Also, proteasomes are kind of enzymes, or I should say lysosomes, that are going to break down large protein complexes. Mitochondria are the next organelle. These are small jelly bean looking organelles within the cell, and the role that they play is to provide energy. This is where ATP are going to be recycled. We talked about how ATP is used as an energy source for the cell, and the phosphate gets broken off and then has to be reattached to it. That recycling occurs in this particular organelle. You will see how the mitochondria takes sugars that you consume and uses the energy within that sugar to attach phosphates back onto ATP to allow that molecule to be recycled. Okay, finally, we'll go to the last structure, the nucleus. Technically, the nucleus is not an organelle. It's a kind of a separate thing within the cell, and it is the housing or repository of your DNA. So all the DNA that you use for producing the proteins of your body largely are found within the nucleus. The composition of the nucleus is that it has an outer membrane around it. We call the nuclear membrane, and within the walls of this membrane there are these large pores that allow substances to move in and out. We'll talk more in the lecture about chromatin and the nucleolus. So I want to just kind of stop there if we can, and I'll either make a second video or we'll pick up from here in lecture. So stay tuned.